Hello viewers. Welcome to the YouTube channel. In this video we will discuss Class 12 or 2nd PUC Biology Chapter 1, that is Sexual Reproduction in Flowering Plants. Part 2. From Basic to Advanced Level for Board, CET and NET. Just subscribe and comment to get PDF. Do support for more updates. So watch till end, don't forget to comment, share, subscribe the channel. Class 12th Physics Class. Video link is given in description, don't miss it. Today, starts with Structure of Anther. Let's shortly revise previous class. Sexual reproduction is the process of development of new organisms through the formation and fusion of gametes. The flower is the main structure concerned with reproduction. The reproductive organs or the sporophylls are produced within the flowers. The sporophylls are of two types: microsporophylls, stamen, and megasporophylls, carpel. Stamen is distinguished as filament, anther, and connective. Carpel is distinguished as ovary-bearing ovule, style, and stigma. The whole process of sexual reproduction in flowering plants can be divided into 3 steps. 1. Pre-fertilization. 2. Post-fertilization. 3. Double fertilization. Pre-fertilization. Structure and events. The pre-fertilization events can be studied under following points. 1. Pollen grain formation. 2. Embryo sac formation. 3. Pollination. 4. Pollen pistil interaction. Note: for CET and NEET, a single vascular band extends from the filament into the center of the connective and continues into the anther. In Malvaceae, the anther is monothecous and bisporaginate. Monothecous anthers are also found in Moringa and Wolffia plants. In Arceuthobium, there is only one microsporangium per anther. This condition is called monosporangiate. Next, structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers. They are: 1. Epidermis. 2. Endothecium. 3. Middle layers. 4. Tapetum. Let's discuss one by one. 1. Epidermis. It is the outermost layer of anther. It is a single-celled, thick, and continuous layer. 2. Endothecium. This layer is present below the epidermis. Structure. It is a single-celled, thick layer. The outer wall of these cells remains thick-walled, but inner walls and radials become thick due to thickening of alpha-cellulose fibers. Endothecium is hygroscopic in nature due to presence of fibrous thickening. It helps in dehiscence of anther. Dehiscence of anther means the process where the anther splits open to release pollen grains. 3. Middle layers. This layer are present between epidermis and endothecium. Middle layer is ephemeral, means short-lived, in nature, and absent in mature anther. The number of middle layer ranges from 1 to 3. The outer 3 wall layers, that is, epidermis, endothecium, middle layers, perform the function of protection and help in dehiscence of anther to release the pollen. 4. Tapetum. This is the innermost layer of anther wall, which acts as nutritive layer. Structure. It is a single-celled, thick layer. The cells of tapetum are initially diploid, but they become polyploid and multinucleate. Because of endomitosis and free nuclear division, tapetum absorbs food from the middle layer and provides nutrition to the microspores mother cells or developing microspores. Tapetum secrete hormones and enzymes. Tapetum layer disappear in the mature anther. The developing pollen grains consume the products of middle layers and tapetum, leaving behind the two layers, namely, epidermis and endothecium. Note: for NEET and CET. Types of tapetum. 1. Amoeboid tapetum. It is found in primitive angiosperms. Example: Typha, Alisma, and Tradescantia. 2. Glandular or secretory tapetum. It is advanced type of tapetum. It secretes nutrients into the cavity of microsporangia and does not store it. Example: Capsella. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Next, anther development. The anther initiates its development in the form of a homogeneous mass of a meristematic cells surrounded by epidermis. It becomes four-lobed and four longitudinal rows of acasporial cells are differentiated. Each of these cells divides to form a primary parietal cell and a primary sporogenous cell. The 1-2 parietal cell divides several times to form the anther wall, and the sporogenous cell divides a few times to form the microscopes or pollen mother cells, PMC. The innermost layer of cell wall in contact with the PMCs form the tapetum, which plays a significant role in pollen development. The layer below the epidermis later becomes the endothecium. Next, microsporogenesis. The process of formation of microspores from a pollen mother cell, PMC, through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells, the microspore tetrad. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Note: for NEET and CET, the PMCs divide meiotically, each forming generally tetrahedral tetrads. Cytokinesis may be successive or simultaneous. Successive type is advanced type. Tetrad are of 5 types. They are tetrahedral, isobilateral, decussate, T-shaped. Linear tetrahedral is most common. In successive type, the cell wall is formed after meiosis 1 as well as meiosis 2, thus an isobilateral pollen tetras is formed. It is a characteristic feature of monocote. In simultaneous type, each nuclear division in microspore mother cell is most followed by cell wall formation. Pollen grains. The pollen grains represent the male gametophytes. If you touch the opened anthers of hibiscus or any other flower, you would find deposition of this yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture sizes, shapes, colors, designs seen on the pollen grains from different species as shown in figure. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent 2-layered wall. The hard outer layer called the exine is made up of sporopollenin, which is one of the most resistant organic material known. Exine can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin is so far known. The inner wall of the pollen grain is called the intine. Intine is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. 1. Vegetative cell. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. 2. Generative cell. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle-shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at this two-cell stage. In the remaining species, the generative cell divides mitotically to give rise to the two male gametes before pollen grains are shed, three-celled stage. In Calotropis and orchids, the pollen of each anther lobe formed a characteristic mass called pollinium. Pollen grains can be monoclopate, having 1 germ pores, bicolpate, 2 germ pores, and triclopate, 3 germ pores. The branch of study of pollens is called palynology. Pollen grains causes chronic respiratory disorders, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Let's finish today's topic here. Don't forget to subscribe and share the channel for more updates.